The overall goal of our macrofoam-based surface sampling method is to identify outbreak etiology, overall evaluation of environmental contamination dynamics of the virus, and effectiveness of remediation strategies for the norovirus.
This protocol detailed how to collect swab sample from environmental surface and how these swabs should be stored and transported, and which laboratory protocol are recommended for the detection and typing of the norovirus from these swabs.
Key improvements of this environmental swab protocol for noroviruses is that we have determined the best type of swab material, how much norovirus can be recovered from hard surfaces, and that the protocol can be used effectively to sample larger surface areas. In this video, two of my staff scientists, Geun and Preeti Chhabra, will demonstrate the swab protocol for the detection and typing of noroviruses.
To begin swabbing, proceed to the sampling area and move the swab across the sampling area with one stroke in the horizontal direction, one stroke in the vertical direction, and one stroke in a diagonal direction. Place each swab into a separate tube and tighten the cap securely. Sample surfaces with stainless steel, sample a toilet seat surface, and sample a doorknob. Next, label one 15 milliliter tube and one RNA MIDI column for each sample. Include a negative extraction control in each experiment. Prepare an empty bottle to collect universal nucleic acid extraction or UNEX buffer waste. To prepare the lysis solution for 10 swabs, mix 25 milliliters of UNEX buffer with 25 milliliters of PBST. Add 50 microliters of coliphate.